Okay, welcome back. So our first invited talk is by Belinda Phipson from the Peter MacCallum Cancer Center. Belinda is one of the authors of the mismethyl bioconductor package, and she will be Belinda will be telling us all about this work today in a talk titled Gene Set Testing for DNA methylation data.
Thank you for the introduction, and thank you so much for inviting me to speak today. So this work that I'm going to talk about is joint work with Jovana Maksimovic, and we work together in the Alicia Oshlak's group at the Peter MacCallum Cancer Center. And I also just wanted to say I absolutely love the BioC Asia hex sticker. I think it's really beautiful artwork. I just love it. So I thought I would start with just a tiny bit of background about me. So I'm originally from South Africa, and I'm a statistician by training, and I moved to Melbourne, Australia almost 15 years ago. And I started working in bioinformatics with Professor Gordon Smythe at the Walter Eliza Hall Institute. And over the course of my bioinformatics career, I've worked mostly in gene expression methods development and analysis. So during my PhD, I developed methods and contributed methods to the Limma and Agile package. And then during my postdoc, I moved to Alicia Oshlak's group. We were at the Murdoch Children's Research Institute, and now we're at the Peter Mac. And I've worked in single-cell RNA-seq analysis, and specifically we developed a simulation for single-cell RNA sequencing data. And I have a package called Speckle in GitHub as well that has some methods for analyzing single-cell data. And I also work in DNA methylation methods development. And Jovana and I developed the mismethyl bioconductor package, I think 5 or 6, maybe longer years ago now. And we have a few different methods in there. And today I'm going to be really focusing on some recent work that we've done about gene set testing for DNA methylation array data. And this was just recently published this year in Genome Biology. So what is DNA methylation? So DNA methylation is the chemical modification of the cytosine C base through the addition of a CH3 methyl group. And it occurs primarily in the context of these CG dinucleotides, and we call these CpG sites. So why is DNA methylation interesting? Well, it's essential for normal development, but it's also disrupted in disease, and it's really, really disrupted in cancer. And another thing that's really interesting about methylation is that it is dynamic. And so even as we age, our methylation profile changes. And if we're able to measure the methylation status of a few CpGs, we can actually predict a person's epigenetic age. And because it is dynamic, it also makes it an attractive therapeutic target. And in normal cells, around 80% of CpGs are methylated, with the exception of these clusters of CpGs, which we call CpG islands. And these CpG islands, if they're in the promoters of genes, they tend to be unmethylated. And there's thought to be, there's a correlation with increased gene expression with these sort of unmethylated CpG islands in the promoters of those genes. So how do we measure methylation? Well, probably the most common way to measure methylation is actually using bisulfite sequencing technology, but microarrays are still a popular choice for doing this. And they're commonly used in large cohort studies like EWAS or things like that. And so Illumina's got an array called the EPIC array, and it can profile almost 1 million CpGs across the human genome. And this array has been really popular, and Illumina's actually just recently released a mouse methylation array. So using these arrays, we get a methylated signal and an unmethylated signal. And so we can calculate what we call these beta values, which is the ratio of methylated to unmethylated signal. And so when you think about the measurement that you're getting, we usually have a sample with thousands of cells. But if we looked at a single cell, we can look at the CpG site on the 2 chromosomes, and it can be unmethylated. So we would have a beta value of 0. The same CpG site could just be methylated on one of the chromosomes and not on the other, and that would be a value of 0.5. And then if the CpG site is methylated on both chromosomes, and the beta value is 1. And so because our sample has thousands of cells, what we tend to see is beta values that are continuous and ranging from 0 to 1, with a peak near 0 for the unmethylated CpG sites and a peak near 1 for the methylated CpG sites. But we also tend to analyze what we call M values. So we tend to do statistical analysis on the log2 ratio of the methylated to unmethylated signal, as this value tends to have a better statistical properties. And so what we're usually interested in is finding methylation differences. So methylation is very cell type specific. So we often study it when we're trying to understand cell types. So for example, looking at different kinds of T cells. There's a lot of data on the Cancer Genome Atlas for methylation arrays, so we can look at cancer versus normal. And the sort of patterns that we're looking at, are we looking for these differences in mean value? So this is an example of just one CpG site. Here are some cancer samples and here are some normal samples, and you can see a clear shift between those normal and cancer samples for this particular CpG. And methylation is also affected by the environment. So the most common example that we have really is the smoking versus non-smoking. So smoking can alter your DNA methylation profile. So how do we analyze this kind of data? So the software tools to find differentially methylated CpG sites or regions are really well established. And there's almost 100 bioconductor packages that analyze methylation data. And so there's a few steps in the analysis pipeline, and we've got quite a comprehensive workflow on F1000 about how to do this sort of analysis. But we start off with pre-processing. So we load the data into R, we clean the data, we remove bad samples, we remove sort of poor performing probes, maybe probes with SNPs in it. And we do a lot of this low-level data analysis with a package called MIMFI, which is one of the most popular packages for analyzing this kind of data. Then we normalize the data, and MIMFI has also got a lot of normalization methods, but Miss Methyl also has a couple. And then once the data is cleaned and normalized, then we would find differentially methylated probes or differentially methylated CpGs, and we can call these DMPs. And then there's other sorts of analysis that you can do after that. You might be interested in CpGs that are different in terms of their spread, not just their mean methylation values. So we could do a differential variability analysis, which we've got a method for that in Miss Methyl. Or you might have thousands and tens of thousands of CpGs that are differentially methylated, and you think, oh, maybe I'll do a region analysis. And so you can find differentially methylated regions, and we like to use a package called Demarcate to do that. And then once you've got like a long list of these differentially methylated CpGs or regions, you probably want to sort of understand what's going on with the biology a little bit more. And so we wanted to do pathway analysis or gene set testing for this kind of data. But actually it's not that simple to do, because what we end up with is a long list of CpGs, and we want to sort of understand the pathways that are involved. And so the issue here is sort of annotating those CpGs to genes and then trying to do some sort of gene set testing that gives good results. So one of the issues though is that gene set testing is biased for methylation array data, and this is due to the composition of the array. And I've got a little example here where we've got two genes. We've got gene 1 has got 3 CpG sites, and gene 2 has got 8 CpG sites, measuring methylation across these genes. And there is one CpG that is shared between gene 1 and gene 2. So the most obvious source of bias here is what we call probe number bias, where the genes have differing numbers of CpGs measuring methylation, and that actually affects the likelihood that you find that gene differentially methylated. And so what we do is we actually, from the data, we calculate the probability that a gene is differentially methylated according to the number of CpGs, and there's often this strong positive correlation that we see. And the second issue is this multi-gene bias, that's what we call it, where around 10% of gene annotated CpGs are actually annotated to 2 or more genes. And the issue with this is that genes that are co-located or they're close in genomic space can also be functionally related. So they might be in the same Go category. And if you treat these genes as independent, then you actually increase your risk of false discovery. So both of these biases increase your rate of false discoveries in terms of gene sets that are enriched. So actually quite a while ago, maybe 5 years ago, I wrote an initial version of GOMETH to account for probe number bias. And so what I'm showing you here is a plot of 100 null simulations using randomly sampled CpGs, ranging from 50 to 10,000. And what we used to do was just do a hypergeometric test where we've associated the CpGs to genes. And so what we want to see is we want to see these violin plots on this red line or below. And with a hypergeometric test, as you increase the numbers of CpGs that you've randomly sampled, you actually increase the proportion of Go categories that with a significant p-value. And so our original implementation of GOMETH was in purple, and we thought it did pretty well, but then we cottoned on to this multi-gene bias issue. And so we've rejigged GOMETH to take into account both sources of biases in the pink, and we can see that there isn't this trend with the number of CpGs anymore. So we thought, after we'd done all this work improving GOMETH, we thought we might as well write a paper about it and actually publish it. And by the time we got around to writing the paper, there were 2 other methods available, 2 other software packages that actually does this. And 2 packages are EBGSEA and methylGSA. methylGSA is a bioconductor package, and EBGSEA is a method that's available in the CHAM package, or the better implementation of it is actually on GitHub. And the issue though is that these 2 methods require the M values, whereas GOMETH you can just give it CpGs to do the enrichment analysis. So what we did was we took some normal samples from the TCGA kidney cancer data set, and we just randomly sampled 5, 10, 20, 40, and 80 samples and put it into 2 groups, and then did differential methylation of these sort of 2 artificial groups where we don't expect to see any differential methylation or significant enrichment of gene sets. And then we calculated the proportion of gene sets with a p-value of less than 0.05. And again, we expect to see these violin plots below or around this red dotted line. And so I guess what's we were very happy to see was that GOMETH was really good at controlling the type 1 error or false discovery rate. And we also noticed that the MGLM method and methylGSA also did a good job. But these overrepresentation methods, this MRRA GSEA and MRRA ORA, we have way too many false positives. So that there's way too many gene sets that are statistically significant when they shouldn't be. And the EBGSEA methods are pretty good. They probably are have a bit too many significant gene sets, but they seem to be okay. So we had analyzed some null data, and then we wanted to also see how these methods worked on a data set with real methylation differences. And so Jovana found this nice blood cell types data set where we've got 6 different blood cell types, and we analyzed, we did a differential methylation analysis of 3 different independent comparisons. So CD4 versus CD8 has got the smallest number of significant CpGs, and B cell versus NK cells have got the most. And so I'm just, for the purpose of this talk, just going to focus on the B cell versus NK cell results. But in the paper, we have everything and another data set in there. And now the issue when you got real data is, how do you know what's true? How do you know which method is more correct? And so we thought quite hard about this, and we decided to define 2 different kinds of truth sets. So the first one was that we took all the immune system process Go-Chow terms and defined those as our true gene sets because it's blood cells. So we thought anything like immune related would be likely to be a true gene set. And then the other thing that we did was Jovana found some RNA-seq data of the same cell types. And we did an analysis of, and we did the exact same comparisons in this RNA-seq data. And then we did gene set enrichment analysis of that data. And then we took the top 100 Go terms, and that formed our second truth set. And then what we did was we ran all the methods, and we ranked the gene sets from most significant to least significant. And we looked at the top 100. And then as we went down the list, we calculated the overlap with the true gene set. So what we want to see is these lines on the top are the methods that work the best. And so what we found was that GOMETH had the most significantly enriched gene sets that overlapped with the two different types of truth sets. And this, again, MRRA, ORA method didn't perform very well at all, but in general, the MGLM method was quite good across all the different comparisons. So I've just talked a lot about CpGs. So what if we have, so that was all at the CpG site level. What if we have differentially methylated regions? So we know that CpGs that are close together are correlated. They're more likely to have the same methylation measurement if they're really close together in genomic space. And so what we do is we can use this software to find these regions where these CpGs are clustered closely together and they have these different mean methylation levels. For example, here is an example of a DMR between colon and lung samples. And there are a number of region finding tools available, and we like to use Demarcate. And then what you end up with after you've done a region analysis is you end up with a list of regions that are statistically significantly different between your two groups that you're interested in. And then once again, though, we have that issue of interpretation. Can we do some kind of gene set testing where we can associate the region with the gene and then do pathway analysis? Again, we're not measuring the actual genes, we're measuring CpGs. And what we found was that region finding is also quite biased. It's very biased, in fact, towards genes with more CpGs. And that makes sense. If a gene doesn't have very many CpGs, then you're not likely to find a region. But if it's got lots and lots of CpGs, you're more likely to find a region, and then it's more likely to, and then you can find a differentially methylated region. But there are no methods specific to, no gene set testing methods specific for this issue. So Jovana thought about how to extend GOMETH for regions. And so what we did was, once you've got your differentially methylated regions, so you've got a long list of them, we can extract those regions and we can look at all the probes that underlie that region, and we can extract all of those CpGs for all of your statistically significant regions. And then again, then we have CpGs, and we can just put those into GOMETH. So we use the statistical framework that we've already developed to do the gene set testing. And so we wanted to know how Goregion performed. And so we analyzed the same data, the sorted blood cell data, Goregion to just a standard hypergeometric test. And we use the same truth sets that we had already defined before. And I'm just going to show you the results for the B cells versus NK cells. And so again, it's the same plot where we're looking at the top ranked gene sets, and we calculate the overlap with our truth sets as we go down the list. And we found that Goregion worked really well. It was much better than a hypergeometric test. And if you want, you can actually we can just look at the gene sets that are in the top 10. And if we look at the output from Goregion, we can see a lot of immune-specific gene sets that are coming up. Whereas if you look at the output from a hypergeometric test, what you can see is a lot of non-specific terms that are not very helpful in terms of interpretation. So protein binding, cell, signaling, these very broad, large terms that are statistically significant, but they don't really give you any insight into the biology. So Anna, so I just wanted to just highlight also, we've done quite a lot of work updating GOMETH in the last year or two, and we do have a couple new features, and one of them is that we can limit the input CpGs by genomic location. So we do have a few gene set testing functions. I'll just mention this. We've got GOMETH, which specifically tests Go and KEGG pathways, but we also have GSA-METH, which is where you can just, it uses the same statistical framework, but you have the flexibility to just input your own lists of gene sets. And then Goregion is the region version of GOMETH, and GSA region is the region version of GSA-METH. And basically with GOMETH, the minimum data that you have to put in is the significant CpG. So if you've done your analysis on an epic array, no, 450K I think is the default, and you have 2000 significant CpGs, you can just do GoMeth and with the 2000 CpGs, and it will run the analysis for you. But you can modify bits and bobs, and one of them is the genomic features. So if you are, for example, only interested in CpGs that are in the promoter, what you can do is you can use any combination of these genomic features, which are defined by the annotation from Illumina. So for the promoter, we might consider that TSS 200, TSS 1500, the 3' UTR, and the first exon that could constitute the promoter. And so what we'll do is that we'll further filter the significant CpGs that have been passed to GoMeth to only include the CpGs within promoters of genes. And then you might be interested, for example, in only the methylation in the body of genes, because they might have there might be a different biological pathways that happen in terms of promoter methylation and body methylation. So you have the flexibility to look at that separately now. And the other thing which many users asked us for is which genes are significant and in terms of driving the significant gene set. So we've also added in a parameter here where you can say sig.genes equals true, and that will just append an extra column to the output that has the genes that are significant and contributing to that specific Go term. So GoMeth, and I'll just summarize, GoMeth and Goregion can test for enrichment of any gene sets. They're very flexible and they're very robust methods. They're based on Wallenius's non-central hypergeometric test, and it can take into account these two different sources of bias that I've spoken about, what we call probe number bias and multi-gene bias. And we've done extensive evaluation and comparison with simulated data, with resampling normals from 2 different TCGA data sets. I showed you the Kirk one, but we've also done, I think, maybe breast cancer normals as well. And we analyzed 2 different types of cell type data sets with real methylation differences, and we found matched gene expression data so that we could create these independent truth sets to help us in terms of our evaluation. So with that, I just really want to acknowledge Jovana, who we worked very closely together on this. Jovana did all of the analysis. I did most of the coding, and we wrote the paper together. And then of course, Alicia Oshlek gave us really helpful feedback on that. Jovana has gone through the effort of making an awesome analysis website using the Workflower package. So, and there's heaps more in the analysis website than there is in the paper. And what I've talked about today is only a small amount of the work that we did to evaluate these functions. And then I just wanted to quickly end with a little plug about myself. So next year, I'm going to start my own bioinformatics research lab within the bioinformatics division at WeHi. And so I'm looking for students, and as well as I'll be hiring a postdoc as well. So if you're interested in any of this sort of work across different kinds of data, I'm very interested in new kinds of data and new kinds of protocol, please get in touch. And thank you for your attention. I'm happy to take any questions.
Thank you, Belinda. If anybody has a question, please leave it at the Slack channel. I will start by a question from myself. So what do you recommend using, like the feature that you just showed at the being added to the package, like selecting the genomic features to limit the significance to what are the biological interpretation or the biological cases of how to assign the CpGs to the genes in general, only to specific features? What do you recommend for people to do, especially the biological interpretation of each case?
Yeah, so we actually, when we're annotating the CpGs to the genes, we just use the Illumina annotation for the array, because they've got quite extensive annotation about what they think these CpGs are, whether they lie in a CpG island, whether they lie in the promoter. So we rely on that. But one of the things I will say with methylation, it's a little bit, we like to say that methylation influences gene expression, but it's not always that simple. And I think the most evidence that we have seen is that methylation in the promoters of genes and CpG islands, there's more evidence for that influencing gene expression. So I think if the promoter is unmethylated, then the CpG island, in the promoter of a gene is unmethylated, then you're more likely to see increased gene expression, activation of that gene, for example. But then there's sort of some papers that say, well, if there's a methylation or whatever in the body of the gene, that there's a different relationship. So there's sort of, it's not the relationship between methylation. What I'm trying to say is the relationship between methylation and gene expression is a little bit murky. And I guess when we're doing pathway analysis, we're assuming that there's something happening at the gene expression level. And so what if you just use GOMETH with the defaults, you'll get usually what we found is you get very sensible results. And what I think the reason for that is that the arrays are actually very biased towards having CpGs in the promoters of genes. And so there's heaps more CpGs in the promoters of genes anyway. So I don't think you need to worry too much about it, but if you wanted to be very sure that the signal you're observing in your gene set analysis is more closely linked to the promoter, you can then sort of use that additional functionality to reduce your set of CpGs. Sorry, that was a really long answer.
Yeah, we have another question here from Stefano. So the question reads, I was curious about the two bias plots about the number of CpGs with statistical significance. So why do we see a step and not more like a linear relationship?
Yes. So we don't fully know what that's from. That has intrigued us as well. And we see that step. So that's for the region analysis. I think that's Stefano is talking about. We see that step in a lot of our plots when we do region analysis, and maybe not so much at the CpG level analysis. And one of the things that we think it might be capturing is this, the sort of because it's an empirical fit. So we don't enforce a particular relationship. It's sort of an empirical fit. We think it's actually taking into account co-methylation as well. So the fact that those CpGs are actually correlated, but we don't fully know exactly what's driving that little bump in that plot.
I would like to thank you again for this interesting talk, and congratulations for the new position. And if you have any questions, you please get in touch with the speaker through the channel or through I see the Twitter handle here. So I'll close this. We will be back in a few minutes.
Thank you. Thank you very much.